All right, welcome everyone. I'm pleased to introduce our first speaker of the semester for the Bair Ranch Seminar Series, Dr. Sarah Moorey. Sarah received her bachelor's degree from Auburn, and then she traveled out to Mizzou, where she received her master's degree under the guidance of Mike Smith. During her time there, she also got to work a little bit with Tom Geary, so she has a little bit of Montana in her background. Then after receiving her master's, she traveled back to Auburn, where she received her PhD under the guidance of Fernando Biazzi. And so please join me in welcoming Sarah. She gives us a talk today on the preovulatory follicle, the master regulator of fertility in cattle.
Thank you, Sarah. And thank you all. I really appreciate your department and the Bair Ranch Foundation for allowing me the opportunity to come here and meet with you. So as Sarah said, today I'm going to talk with you about the preovulatory follicle and its role as a master regulator of fertility in cattle. But first, I think it's important to take a few steps back and really focus on why fertility is important. So this is just a table that shows the estimated annual loss in $1 million for some conditions that beef or cattle producers face. And as you can see, reproductive failure is right at the top of this list, causing an estimate of over $2,500 million each year. Now, when we think about reproductive failure, the first thing that comes to my mind is having open cows at the end of the breeding season. However, interestingly, breeding season pregnancy rates are actually quite high. So if we look at this table, we can see that regardless of the breeding season length, cattle actually have quite good fertility by the end of the season, and over 90% of cattle are pregnant. However, where we have reproductive inefficiency lies within pregnancy rates to a single service. So when we think about beef cattle, we could expect only about 50 to 65% of our cows to become pregnant to a single service, be that a natural service from the bull or to artificial insemination. And this number drops significantly further when we consider high-producing dairy cows. Dairy cow fertility can be lower than 30% per service. So pregnancy rates to a single service are a huge issue for the economic viability of the cattle industry. Now, as you all are aware, I know there's a lot of people in this room that are not reproductive physiologists. There's a lot of things that can influence pregnancy rate. And I'm first going to talk about nutrition. Because of our audience, I know that you're aware that nutrition can have a great impact on reproduction and fertility in cattle, and that a cow's body condition or her metabolic status when she is inseminated can influence if she becomes pregnant or not. There are also numerous other factors, though, that influence fertility, such as the ability of the conceptus membranes to form a highly functional placenta to support pregnancy throughout until gestation. Overall health of the animal. Though our cows don't suffer from COVID-19, we do need healthy animals for optimal reproductive efficiency. Furthermore, we need to have maximal fertility in our sires. We need a uterine environment that is receptive to pregnancy. And we need efficient inseminators or semen handlers in technologies that do utilize human manipulation to allow for pregnancy to occur. However, today, I'm going to focus on the preovulatory follicle and its huge role in pregnancy establishment, specifically through the ovulation of a highly competent oocyte for embryo development. The preovulatory time period is essential for fertility. Regardless of any other parameters, the ovulation of a competent oocyte from a follicle that will turn into a functional corpus luteum is essential. We cannot get a pregnancy without these two factors. And the preovulatory period highly promotes both of these essential factors for pregnancy. So when we think about this, this might be the follicular phase of the estrous cycle that you remember from your undergraduate repro class. That would be from the time of prostaglandin administration or secretion, which would lead to luteolysis, until ovulation. And we can further divide this period into a proestrus period, before estrus, before the ever important LH surge, and estrus, or post-LH surge development from the surge to ovulation. Now, proestradiol pre-LH surge development of the follicle represents a huge increase in that follicle's functionality. As you can see here, the follicle is growing in size. It's producing more and more estradiol, which will eventually allow the animal to exhibit estrus and experience the LH surge. The LH surge then acts as a stimulus to lead to ovulation. So this would lead to luteinization of the follicular cells, breakdown of the follicle wall, increased angiogenesis in the follicle, and oocyte maturation that all occur during this ever important time. So when we think about this, we could think of proestrus, this period, as a get ready phase. And then the LH surge as our go marker. And then this period from the LH surge to ovulation is really running the final leg of the race after we've prepared. I thought it was very interesting. In 2021, Marc-André Serard published a review paper that talked about follicle development and the ever importance of these later stages of follicle development. And he compared follicles going through this stage to a ripening of an apple. So when we think about apples growing on a tree, we can have a fully formed apple that if we wanted to, we could pick and eat, and it would be quite unripe. So a green apple that's not ready for picking, we could eat it. It probably wouldn't taste good. It's not optimal. And then as that apple continues forward, it begins to ripen. And then finally, only in the last days of that apple's time on the tree does it fully ripen and create an apple that we would really like to eat. So he likened this to follicle development. And during this time, when the follicle is increasing in its functionality and preparing for the LH surge, those would be those final days of the apple ripening on the tree or the follicle ripening on the ovary. Now, within the follicle, there are multiple somatic components that greatly influence the oocyte, as well as the follicle's ability to create a pregnancy. When we think about the oocyte, one follicular oocyte relationship that might first come to mind is between the oocyte and the surrounding somatic cells that are called cumulus cells. And these cumulus cells commute with the oocyte both directly through gap junctional communication, which you can see here and here, that actually span the zona pellucida and transfer molecules such as RNAs, metabolites, or amino acids directly to the oocyte. And the oocyte and granulosa cells can also signal to each other through paracrine action. We'll talk a little bit later as to why these connections are so important, but I just wanted to set a stage that the cumulus cells are definitely a nurse type cell that can send the oocyte molecules that are essential for fertility. Further, when we think about the follicle, which would be outlined here in pink, it goes beyond just the cumulus cells and the oocyte. The follicle is surrounded by granulosa cells, which are the primary steroid hormone producing cells that are within the follicle, along with the theca cells on the outside of the follicle wall. And then the follicular fluid that consists of fluids and metabolites that are products from both these granulosa cells, the cumulus cells and oocyte, and then input from maternal circulation that can cross through the blood follicle barrier. So if we look down here, you can see that there are numerous products within this follicular fluid that could be quite useful for cells that are metabolically active or are undergoing a type of differentiation. We can see glucose metabolism type components, nucleotides, amino acids, and many other products that I hope at the end of this talk you'll be convinced are essential for optimal fertility. So going back to Dr. Serard's ripening of an apple, for the rest of today's talk, I really want you to focus on that we're having this follicle ripen and prepare for the LH surge and ovulation, and that as that follicle progresses to this point, time is running out. Time is running out to prepare itself for optimal fertility. This is really the follicle's last chance to gather everything it needs to perform and become an optimal corpus luteum and ovulate an optimal oocyte. Now, it's very interesting that we talk about these final stages of follicle development as being essential. When we think about improvement in the livestock industry, I always focus on reproductive efficiency, but genetic improvement gives us so much opportunity to improve our industry. 3 common genetic improvement technologies, such as in vitro embryo production, multiple ovulation embryo transfer, and fixed time AI that we'll talk about later, manipulate the follicle's development and either modify the preovulatory follicle composition or reduce or eliminate the time that the oocyte is exposed to that critical preovulatory follicle. The most drastic of these 3 reproductive techniques for genetic improvement is in vitro embryo production. So as you can see here, Trish's clicker bothers me. I feel like I think between each slide, which button do I push? Push the right button. As you can see here in vitro embryo production, these oocytes are taken from follicles that are actually quite small in a follicular way. They are then brought into the laboratory, washed and sorted, and undergo in vitro maturation before they undergo in vitro fertilization and downstream embryo development. So as you might imagine, the oocytes from these in vitro procedures actually have quite reduced competence for development of a blastocyst in future pregnancy. This table just shows us that indeed in vitro matured oocytes, shown here in white, do have lower blastocyst rates than do their in vivo matured counterparts. And this difference is due to a multitude of factors. Like we said, many things influence fertility. However, one driving factor of these reduced blastocyst rates and reduced oocyte quality is the fact that these oocytes are never exposed to a preovulatory follicle. They're not from animals that experience proestrus nor estrus, and they do not have access to these essential metabolites or signaling molecules that are accrued during those essential time periods. Multiple ovulation embryo transfer also gives us large variability in the oocyte's competence to develop into a quality embryo. And here, as you can see, multiple follicles are allowed to be selected and continue on through dominance and preovulatory development. So these follicles are exposed to the preovulatory conditions. The animal will undergo luteolysis, proestrus, have an LH surge, and then ovulate. We know that. We breed these cows that are superovulated, just like we'd breed a cow undergoing fixed time day I. However, these 5 follicles are not all at the same stage of their follicular development whenever the multitude of estradiol production from all of them triggers the LH surge in their dam. So when we think about that, these follicles have reduced duration of their exposure to the preovulatory follicle. Interestingly, even though this follicle may have reduced maturity compared to this one at the time of the LH surge, the LH surge actually happens earlier for all of these than it would have happened if only one preovulatory follicle was developed. And that's because of that combined estrogen production. So each of these guys is producing estradiol, and it's actually a little bit less than just the one follicle would produce on its own. So some studies have shown that their intrafollicular estradiol levels have a variation from about 400 to 600 nanograms per mil, whereas a single large preovulatory follicle can produce over 1,000 nanograms per mil. But when we get 400 times 5, as you can see, we'd reach a threshold for ovulation much earlier and cut the time that these follicles have for preparation or for ripening early. Now, I also thought it was interesting when others have looked at these oocytes. So we know from cattle that fertility is variable and that oocyte competence is influenced. Others that work with rodents, though, have gotten a little bit further into this to see why or how oocyte competence is influenced, which is really quite funny to me when you think about super ovulating a rodent. But in these marine studies, oocytes that were from superstimulated ovaries actually had less ATP and then the poor blastocyst development that we'd expect. So keep that in mind, if you will, that ATP or metabolic competence of that oocyte is indeed influenced in this multiple ovulation embryo transfer scenario. And then finally, fixed-time artificial insemination does manipulate the preovulatory follicle. Now, granted, this is not to the level that we would see in multiple ovulation embryo transfer, and those follicles definitely are allowed to progress through preovulatory follicle development. As you can see, these cows have a proestrus period, and if you've done fixed-time day I in a herd, you probably know that about half of them will indeed express estrus. However, in animals that do not express estrus, we give an injection of gonadotrophin-releasing hormone at the time of insemination. And this will lead to the LH surge rapidly following GnRH administration. So this LH surge is induced prematurely, and therefore, again, we are cutting off the amount of time that that follicle is allowed to ripen and progress towards when it would stimulate its own LH surge. Now, before I say anything else, I want to make it very clear that I think fixed-time day I is an awesome technology. I'm not standing here showing you this data to tell you that fixed-time day I is not good or is not beneficial for a producer to utilize. So when we look at this, you can see that this clicker, you can see that within the herd, across animals that are cycling and even animals that are in estrus, when you start your breeding season, you can utilize fixed-time day I. This was quite a successful study or group of studies, but to get roughly 50% of your animals pregnant on the first day of the breeding season to an AI sire. So this is very useful technology. However, there's room for us to improve it. If we can learn more about what we do during this time period by having this induced ovulation to influence the follicle's quality and thus pregnancy rates in the oocyte. So when we think about that, we really need to look at the follicle's physiological maturity. And that's where my lab and my focus comes in, because we would like to learn what makes a physiologically mature follicle more optimal to support future pregnancy and oocyte developmental competence. Now, there's two ways that thus far the model that we use has defined physiological maturity of the follicle. That would be the follicle's estradiol production. So when we think about that, as the follicle's going towards stimulating the LH surge and estrus, it's making more estrogen. So if a follicle has more estrogen when we induce ovulation, it was further on its way to its end goal. It was riper. We can also look at the diameter of the follicle, because we know that follicle is growing in size as it progresses through that time period as well. And Tom, Gary, and Mike Smith, George Perry, and many others, which I'll show you some data in a minute, have done quite a bit of research and really were at the forefront of discovering these relationships and helping build this model for future study. So when we think again about those physiologically less mature follicles during fixed-time artificial insemination, they've got reduced exposure to the preovulatory time period, and they have reduced oocyte competence for embryo development, which we'll look at in just a minute. But before we look at that, I want to start at the top and show you guys the pregnancy data and then work down towards the oocyte. So looking at the pregnancy data, which again was from Tom, Mike Smith, and George Perry, you can see first of all that animals that ovulated from estrus, so these animals displayed estrus within the protocol, they did not have an induced ovulation, their pregnancy rate was higher than animals that relied on GnRH to induce ovulation. We'd expect that. That follicle is more mature. That's a more ideal follicle. Furthermore, though, when we look into these animals that were induced to ovulate, you can see here that animals that were induced to ovulate a very small follicle or a smaller follicle had drastically reduced pregnancy rates at day 27, and then increased embryonic mortality to have further reduced pregnancy rates by 68 compared to their counterparts that ovulated a larger, more mature follicle. This is just some additional figures from other studies to show you that's not unique to data that was produced here in Montana at Fort Keogh. This is across the world with Taurus and Indicus cattle. The same relationship with the follicle size or status and fertility shown in pregnancy rate exists. Now, after this difference in pregnancy rate based on follicle status was determined, a series of studies was performed to try to figure out exactly why. What players in the fertility game are impacted? Because really it could be anything. We think about this follicle ovulates an oocyte that could be impacted. Future corpus luteum could be impacted. As we see here, based on progesterone of large and small follicles after induced ovulation, it indeed is impacted. Furthermore, the oviduct would be influenced by the maternal physiology environment created by the follicle, and then uterine receptivity to pregnancy, ability of the conceptus to elongate and create a quality pregnancy could all be influenced by preovulatory status. Today, I'm going to focus on the oocyte. So I think Tom came to you guys back in October and he focused on the uterus. So it set it up really nice for me to get to say, as he said, it's very complicated and I'm not going to talk about it today. So watch his recording if you're interested to learn about the uterus. Today, we're going to talk about the oocyte. And indeed, when they studied this, oocytes from follicles that had a smaller diameter had reduced probability of fertilization and reduced probability of creating a transferable embryo by 7 days after insemination. So this is just the experimental outline of that study published by Atkins, and then Jinks also looked at estradiol. So as you can see, they synchronized cows, as we would commonly do in a fixed time day eye procedure, started with GnRH, 7 days later prostaglandin, 2 days later at the time that viewed AI, they gave GnRH and measured estradiol concentration in serum, as well as the follicle's diameter. 7 days later, single embryo flushes were performed, and they determined if there was an influence of follicle diameter and estradiol on embryo quality, which we've already said there was. And this really is the first stage of preliminary data that led to the overarching hypothesis of my lab. And that's that changes in the preovulatory follicle's composition leading up to the LH surge are physiologically essential because they directly support oocyte developmental competence for pregnancy. So the first paper that we worked on to really get to the meat of this, Tom's shaking his head, started when I was actually just a master's student, studied with Dr. Smith at Missouri and Tom Gary, and we performed synchronization in a group of cattle for the same synchronization protocol. But at the time that you'd normally perform timed AI, we didn't inseminate those cattle. We waited 23 hours later, and we performed preovulatory follicle aspiration to collect the contents of the follicle. So the follicular fluid, the oocyte, the cumulus cells, and the granulosa cells. And then we performed RNA sequencing on the oocytes and the cumulus cells from those follicles. I had to put this picture up here. Sarah said I had roots here. This was from the, I guess, first week I spent in Montana. I'm the skinny one right here. I don't know what's happened from then to now. But that was a great summer and really helped me begin my career through that master's degree. So when we look at the transcriptome data, just showing you a heat map of the two end results that were the most impactful. You can see here in the top, this is within the cumulus cells. We had significant enrichment in the glycolytic pathway for differentially expressed genes between or among small, so follicles that didn't display estrus and then were small in diameter, large follicles from cows that didn't display estrus but were larger in diameter, and then spontaneous or estrus follicles, which this would have been the cows that displayed estrus. We can see that abundance for these key glycolytic enzymes, transcripts, increased as follicle maturity was greater. Furthermore, when we look at the oocyte, we see a similar trend. Rather than glycolysis, we've got oxidative phosphorylation. And when we look at the nuclear transcripts, so here up, we can see the same tendency that increased abundance of nuclear transcripts related to oxidative phosphorylation increased as did follicle maturity. Now, it's really interesting. I say nuclear transcripts because we had a number of mitochondrial transcripts. So these are from the mitochondrial genome that actually were included in our sequencing data. They were higher in the small follicle oocytes. So we're still really not sure what's going on there, but I think it's important to chew on and for you guys to see. So when we look a little bit more detailed, glycolysis was not our only enriched pathway. We've got a number of other metabolic type pathways, and our most influenced comparison was between the small and large follicle cumulus cells. Multiple pathways, primarily these glycolytic enzymes. And then also I thought it's important to show you though they didn't pop in our pathway analysis, we've got a number of solute carriers. So these would be the transporters that allow for the uptake of glucose, amino acids, or fatty acids. So we've got 2 glucose transporters that were higher in large versus small. And amino acids transporters or fatty acid transporters. I'm not sure which way these 2 were. I've forgotten it. That's terrible, but I'll be honest with you. But again, metabolic support, they're upregulated in a more advanced follicle. Now, this makes a lot of sense because when we think about metabolism of the cumulus oocyte complex, like we saw earlier, those cumulus cells are tightly connected to the oocyte as it prepares for maturation. And they are actually the driving forces of glucose metabolism within the cumulus and oocyte complex. The oocyte does not have the enzymes needed to directly metabolize glucose. So if an oocyte is going to use glucose, it's going to have to come through metabolic activity from a different cell type. And that's these cumulus cells. So from our seq data, we've got increased transporter of glucose into the cumulus, and then increased abundance of multiple enzymes within glycolysis or for the conversion of pyruvate to lactate. Both pyruvate and lactate can then be transferred to the oocyte, and they can enter oxidative phosphorylation in the oocyte to generate energy through the mitochondrial respiratory chain. Just looking back at this oocyte again, we see that mitochondrial respiratory chain, oxidative phosphorylation, as we said, to be the most influenced by follicle status. Though we did not have enrichment of pathways within the small versus large comparison, there were a number of nuclear mitochondrial oxidative phosphorylation genes that were upregulated in large versus small. And we also have this oxidative phosphorylation pathway in small versus estrous or spontaneous follicles. So there's a really strong trend from this seq data. And I think it really served the purpose of RNA sequencing data. When we did the study, we knew there's something going on, but we really didn't know what it was. After we analyzed these results, we were pretty sure there's something metabolic related to the follicle status. And that has been the focus of my independent program at Tennessee since I started there 3 years ago. We've really been working to dive deeply into the oocyte and preovulatory follicle metabolism. I've gained some great collaborators at UT and Lynette Edwards and Neil Schrick in the animal science department, and then also through Sean Campagna. He's in the Department of Chemistry, but he also runs our metabolomics core, which is where all my samples would have gone through. So I'm going to show you now results from some of the studies of 3 of my past master's students. That's Casey, Emma, and Emma. And then these studies will serve the base for my 2 current students' work as we move into the future. I also want to take this time to share some exciting news about the future. I got an email 2 weeks ago, and we were funded for a USDA NIFA grant to continue our research in this area. So I'm very excited about that opportunity to keep moving forward. So our first key area that we wanted to determine was if follicle status before the LH surge related to oocyte metabolic capacity. And then we were curious at the potential causes for this relationship. The seq data suggested it, but is it real? So we performed a study very similar to the design which you've seen before. Synchronized cows, when we normally do fixed timed AI, don't AI them, check their follicle size and serum estradiol to see the follicle status. and then collect the preovulatory follicle contents 18 hours later. The first thing that we noticed was that ATP is definitely influenced by estradiol production of the follicle. So as we can see here, this is a figure that shows us serum estradiol and then ATP in picograms within the oocyte. We have a positive relationship between serum estradiol, what would have been the time of AI, and ATP abundance in the oocyte 18 hours later. So that shows us increased metabolic preparation for that oocyte during maturation, and then for fertilization, cleavage, and embryo development. And that becomes important because the oocyte requires itself to store a lot of energy for fertility to be optimal and for pregnancy to occur. Oocyte processes of maturation, fertilization, cleavage, and early embryo cell divisions, as you might imagine, require a lot of energy. Interestingly, too, though, as we said, the oocyte can't directly metabolize glucose. Well, neither can the early embryo. So until the embryo reaches about the blastocyst stage of development, they're not able to metabolize glucose directly. They rely on cumulus cell glycolysis when they're still within the follicle and metabolic support, metabolic preparation of the oocyte during that time period, and then some pyruvate and lactate uptake as we continue forward, but this would be minimal. This slide is just to remind you guys of the importance of that cumulus oocyte relationship for metabolic competence, and then to focus on the fact, okay, we have this hypothesis that the cumulus cells have more glycolysis, help support the oocyte, but what about everything else? This cumulus oocyte complex doesn't exist in a vacuum. It's in a biological complex of the follicle. So what about the follicular fluid? We know that all of these metabolic compounds are in it. Does the follicular fluid better support the oocyte and the cumulus cells in a more ideal follicle? And interestingly, we found a renowned metabolic signature once again. So we performed mass spec on the follicular fluid from follicles that either were larger or smaller in diameter. So that would have been our first study. We didn't have hormone data yet on these, but we wanted to go ahead and get this published and get it out because it was really renowned. We identified 38 metabolites within that follicular fluid, and 29 of those had a positive relationship with follicle size at GnRH. And these metabolites were enriched within pathways such as glucose metabolism and amino acid metabolism. Now, moving forward, we got the hormone data, so we were able to include estradiol. We also took a better panel of metabolites and were able to identify 90 metabolites rather than 38. And we saw again 22 metabolites that differed according to follicle estradiol. These all increased with increasing estradiol. And again, we see glucose metabolism, TCA cycle, and amino acid metabolism as key signatures of these different metabolites. This is just a figure to show you some plots. This is data from follicle diameter, but these would look very similar if we'd plotted estradiol. We just didn't plot for the paper, so I didn't have them readily available to show you. But as you can see, alpha-ketoglutarate, glutamate, aspartate, pyruvate, I picked these guys because they're going to be key metabolic compounds and or byproducts of glucose metabolism in the TCA cycle. Indeed, they increase in abundance in the follicular fluid as follicle size increased. And when we put this together into a model that utilize what we know about follicle metabolism, what we learned from the seq results, and then what we've learned from these metabolite results, we can really create a story that there's increased metabolic support of the oocyte in these more advanced follicles. So we think about this glucose abundance within the follicular fluid. We've got better uptake of the glucose abundance through increased glucose transporters, increased glycolysis, so increased production of pyruvate in the cumulus cells of those better follicles. This pyruvate then could directly enter the oocyte, or it could go through the TCA cycle in the cumulus cells, which would then generate numerous metabolic byproducts, which could be shuttled to the oocyte to support the oocyte, or shuttled back out of the cumulus cells into the follicular fluid. This would go both ways. And again, we'd see this in the surrounding granulosa cells. They're not on this figure because they're not directly tied to the oocyte, but increased metabolism of those granulosa cells would again shuttle more metabolites into the follicular fluid, which could be uptaken by the cumulus and sent to the oocyte. So it's really this positive, continuous type factors in the follicle that creates a superior environment and allows us to maximize fertility. So I got curious. This was nice. We had continuous data. But what would happen if we broke our continuous data into categories? So if I have categories, I can apply treatments, high and low estradiol. So we did that, minimal numbers. I didn't expect us to see much. But when we look at clustering of individual follicular fluid samples of either high or low estradiol quartile based on metabolome profiles, we had fully independent clustering of these two groups, which is really renowned for me that the follicular fluid composition of our better and not as good follicles differ completely. There's not overlap here. I'm really excited about that and looking forward to further exploring that and applying some treatments with our USDA grant with my future students. Now, moving forward, up to this point, I hope that I've shown you data to convince you that follicle status at the LH surge is very important and that it can influence factors in the oocyte that occur close to ovulation. So it's setting the stage for oocyte maturation, which would occur during this time period. However, what we don't know up until now is what happens within the follicle. So how does the metabolome profile of the follicular fluid or the metabolism of the follicular cells change during this period of oocyte maturation? This is hot off the press in prep. We performed a study a couple years ago in which we aimed to answer just that question. We synchronized a group of cows. We used estradiol animals in this study because we wanted to look at these changes in an optimal follicle. So we checked estrus on these animals every 4 hours until we observed standing estrus, and then we were pretty much out there continuously. Then we collected the dominant follicle content, so the fluid, the cells, the oocyte, at either the onset of estrus, 10 to 12 hours after the onset of estrus, and then 18 hours after the onset of estrus. And this really allowed us 3 different samples from critical time points in oocyte maturation. The hour 0 samples would have been before the LH surge. We'd have an immature oocyte. This is the get ready stage. The 10 to 12 hours, this would have been post LH surge. The oocyte would have been approaching the stage of metaphase 1, starting to dissociate from the cumulus. This is an intermediate stage. And then 18 hours, we would have had post LH surge again, an oocyte approaching maturation, so approaching metaphase 2. We've kind of run the race at this point. And multitude of samples. Not going to show you RNA sequencing data today. We just got it back a few months ago. I've not had time to analyze it. We're also really excited about digging into the extracellular vesicle mRNA, microRNAs in these samples. But today, we're going to focus on the metabolome analyses of the follicular fluid. And you would expect this to change, right? So everybody thinks it'll change. I'm going to see if I'm the only dumb one in the room that it changed differently than I thought it would. So indeed it changed. We had our good metabolic panel. We identified 90 metabolites. 20 of those differed in abundance based on time post-estrus onset. And I was just fascinated in the fact that the levels of these metabolites, 17 of those 20, increased in the follicular fluid from estrus to 11 hours post-estrus. Then, when we look at the majority of these samples, they either tended to remain elevated at hour 18 versus 0. That's not shown. 5 of them in this top row dropped, such that there was, in this bottom, where am I at? I don't know. This top row shows us metabolites that dropped in abundance from hour 11 to 18, such that they were again lower at 18 than 11, but there was no difference between 18 and 0. And then some others remained elevated such that there was really no difference in either type. You might argue that these are on their way down, they've just not gotten there yet. There was one metabolite, that's aspartate, that did what I thought they were all going to do. It rose throughout the follicle and oocyte maturation period. And this actually makes sense because when you look at aspartate, it's involved in progesterone signaling in cells. And we think about those luteal cells, which the granulosa cells will become. They're going to start becoming secretory of progesterone. This might have a role here. There's also been some studies that aspartate can assist with sperm function. So when we think about this follicle ovulating, we've got more aspartate. Those sperm may be influenced by the follicular fluid. Then we've got a few that just maybe drop from one point to the other and then rose again. But looking at the metabolites that differ, and predominantly they're going to have this up-down signature. We see these same players that we saw in my follicle maturity data. We've got alpha-ketoglutarate, pyruvate, aspartate, glutamate. We've got two new players in lactate and cysteine, but I can still plug them in to this metabolic cycle, even though they're not on the board from our previous results. So the metabolites that are essential for fertility, higher in abundance in better follicles, are changing in abundance as oocyte maturation commences and as that follicle prepares to ovulate. And I just thought this was so interesting and scratched my head. Why in the world didn't I think that this would happen? But I didn't. So when we use that data and perform pathway analysis, as you might expect, we had very similar pathways to what we saw in our follicle maturity data. So any pathway that has a green or blue star was also enriched in the estradiol or follicle size metabolome analyses. And again, you're seeing that the metabolites that contribute to pathway enrichment are those glucose metabolism metabolites. So we've got a story here that really has emerged. And then I'm going to try to tell you what I think contributes to the rationale of these metabolites rising to hour 11 and then going back down. So when we think about back to those three critical time periods, hour 0, the onset of estrus, we're going to have granulosa cells that are undergoing normal cellular processes. So they're going to be using metabolites from the follicular fluid and then contributing metabolites back out. Just normal, regular day. The cumulus oocyte complex is going to be intact. We know our immature oocyte is going to be accruing metabolic substrates and preparing for maturation. So the cumulus cells, just like the granulosa, using our metabolites from the follicular fluid and then shuttling byproducts back out into the oocyte. However, that LH surge creates a completely new response in the follicle. So by hour 11, we've responded to the LH surge. We are responding such that there is increased metabolite and amino acid availability within the follicle to help fuel this response. We require a lot of energy to have the response to the LH surge, to have the cellular transformations. So it makes sense that these cells would need additional metabolic support to do these things. I found a really interesting paper on granulosa cells that actually about this time after the LH surge, they're going to increase their metabolism and amino acid production. So they're doing this functionally. I would imagine the cumulus are too, to help prepare that oocyte for when it disconnects. Also, during this time, we're seeing that angiogenesis and increased vascularity to the follicle. So we're going to have increased permeability of the blood follicle barrier, which is what keeps things from the blood out of the follicle. So if we have a blood follicle barrier that allows more metabolites into the follicle, it would be logical that we would have increased abundance of these metabolites within the follicular fluid at this point. And when we think about that, we've really got a functional supportive role of this. The cumulus oocyte complex is intact, so the cumulus can take all this excessive amount of metabolites and send it to the oocyte in its last chance to store these metabolites before it continues forward. The cumulus themselves are preparing to expand, which is going to, again, be metabolically challenging. So they need these extra metabolites. The oocytes preparing for maturation, again, we need more metabolites. Back to that paper that I saw about cumulus cells, by hour 18, which would be our final stage of metabolism that we collected follicles from, granulosa cells begin incorporating these amino acids that they previously produced into proteins. So So when we look here, they're still performing their metabolism, sending metabolites out, but all of those amino acid metabolites, they're going to be uptaking them and producing proteins out of them to help them for their future role in luteal function as luteal cells. Furthermore, at this point, the cumulus have expanded. We don't have that extreme need for energy, and the oocyte's reaching maturation. So it's kind of stored what it's going to have by this point. So it would make sense that the follicular fluid that bathes and feeds all of these cells wouldn't need to have as much availability of amino acids or metabolic substrates. So I just think that's really cool how the follicle in itself creates this environment to support the oocyte. And I think it will be really neat as we move forward to learn more about this environment and how we can manipulate it either through systemic treatments or manipulate conditions in the lab to help create this profile of metabolic support to the developing oocyte. With that, I had to put a cute tease on this. I hope you've seen in the last 45 minutes that the preovulatory follicle really does have a role as a master regulator of fertility, kind of at the top of our hierarchy, and it works through secondary aspects of its functionality and proper interfollicular dynamics, both pre and post the LH surge, to create hormone profiles and metabolic preparation that leads to improved uterine preparation for pregnancy, superior metabolism of the follicular cells in oocyte, and increased oocyte competence for embryo development, what leads to our end product of increased fertility. And with that, I just want to say a brief thank you to my current and past lab members. Without them, we definitely wouldn't have gotten these studies performed. Also appreciate my collaborators at Tennessee, and then also at other institutions in Mike and Tom. I appreciate the research centers at the University of Tennessee. We've really got a great set of directors and staff at our centers. This is Walt Hitch and Kevin Thompson, who manage the centers where the Tennessee studies I showed you were performed. I appreciate USD and NIFA for funding through my multi-state and now my grant. Also the University of Tennessee Ag Research in our department for startup funding. And we'll end with a great thank you to you guys here in the Animal Science Department at Montana State and to the Bair Ranch Foundation Seminar Series for this opportunity. I really appreciate it. And at this time, I would be very happy to take any questions. And I would also be very happy to steal your excellent lab grad students to join my lab as a research associate. I think I'm hoarse.
Have you seen by NP metabolites that are affect
Right. That's a really good question. And my answer is somewhat no, but I'll tell you a little bit about what we have seen and what we think. So we do know, like we said, with that blood follicle barrier, what is in systemic circulation can enter the follicular fluid. So if you're thinking about metabolic byproducts like pyruvate, glucose, these amino acids, if they're small, so about less than 500 kilodaltons, and if they're neutral or kind of a slightly negative charge, they can pass. So if you've got those type of molecules that are being produced based on nutritional treatment, I think it would make tremendous sense that those would enter the follicle and could influence that follicle composition and lead to these exact changes in animals that are of obese, thin, negative energy balance. Now, we did perform a study. We got really lucky with our large study in that we had some thin cows, we had some of the fattest cows I've ever seen, and we had some moderate cows. So we collected some serum samples and follicular fluid from those cows. And the serum was, I knew this was going to come, so I was prepared. Chris's thing's going to make me look stupid. We had tremendous difference in the serum metabolome. If you're interested in this, we've got that paper published in JAS. but just tremendous glucose pyruvate, TCA intermediates. We think about that cow breaking down fat, breaking down muscle because she's under metabolic stress. That's elevated in the circulating serum. Now, when we looked at the follicular fluid, we didn't see any significant differences in these metabolites in the follicular fluid. However, these cows were fairly short postpartum. They'd have been about 60 days postpartum when we collected the samples. So we don't think that there'd been enough time for this to continuously be going on to get that follicular fluid at that level. So we're further exploring that, also looking at some oxidative stress, that this might generate reactive oxygen species that would pass that barrier and might have that negative impact. So I'm so glad you asked that. Back. Man, I bored you all. This is bad. Tom's being nice to me, doesn't want to embarrass me by asking a question.
I'm not trying, I don't want to. Questions, I have the one-on-ones. I'll ask you a question about, because when you started out and you started talking about superovulation, talked about we're ovulating those oocytes earlier than they would. And there's positive and anti-estrogen had short-term use to delay surge and use it with 20 hours later to get more follicles and
That's a really interesting proposal.
Because Abby and Carolina stayed at home so I could come here, breed cows that were ovulating.
Right.
Next week.
Right. Yeah, Tom, when you say that, I get really excited. I'm like, oh, this would be cool. From a base level, it makes sense to me. We would reduce that systemic estradiol, elongate that period, and in this instant, induced ovulation would be good because we had a longer period. I don't know if there could be potential negative aspects. I'm not as well versed on how that specific compound would work. Where does it function? Would it produce follicular estradiol, or it would just reduce its bioavailability systemically? Like it would get rid of it quicker.
Work on the estrogen receptor.
Okay.
So they're really receptor antagonists.
Okay.
Active or work.
Mm-hmm.
But if you're taking those out.
Doesn't matter. Right. That would
I
be.
think the uterus is important, Sarah.
Oh, it is. I'm just not smart enough to do it and the oocyte. But no, I think that would be interesting, Tom. And I think it would really be a cool way to get at some questions I have with estrogen in the follicle and the oocyte. I have a hypothesis that a follicle that creates more estrogen is just a more functional follicle. I don't know if the estrogen's directly influencing the COC or the cells, or if it's just an indicator of a better follicle. So if we did something like that, where we don't have receptors for that estrogen, we could really kind of piece that out. I think it would be quite neat. But I don't know if it would help or not. I think it's worth trying. Call me when you do it. You want your oocytes, but I want them too. Sarah.
Based on all your data, I'm thinking an IVF land not incorporate factors in maturation. In fact, most people.
Right.
So it seems like suggesting we should be keeping that because we use the house-derived O6. Saying the follicles are certainly not much. I know they're not. I see
Right.
everything that is. So taking a deeper look at what we're maturing things with.
Right. No, I'm glad you mentioned that because I think we should. And I think when we look at this data, kind of following this trend and thinking about all of these essential metabolites, I think this shows us a lot of room for improvement with our in vitro procedures. Goodness knows I fight and try to stay out of in vitro land, but I think the data really supports it. And I think it's a unique data set that a lot of improvements could be made for in vitro maturation. If we could try to better characterize this, better manipulate this in vitro, maybe we take them out of the dish 10 hours after, swish, swish, let's add some metabolites so that they've got that extra support. I think that this is the area that this research will make the fastest translational impact. I think we've got huge translational potential in the cow, but me or somebody way smarter than me is going to have to figure out that blood follicle barrier, how we can manipulate the follicle. Whereas here it's in a dish, we can manipulate it and get started on it. I'm glad you said that too. You get me excited. We talked about this, and I was trying to go fast, and I didn't talk about this, but this myoinositol actually has huge roles in fertility. It's integrated into calcium signaling type pathways, and they actually supplement women that are having fertility issues with myoinositol to improve fertility. And I know that some in vitro labs will use myoinositol in their media. And I know you had said you weren't familiar. So I think even, yeah, I'm glad you did. So I think even beyond just my huge metabolic scheme, there's other molecules in here that we've got to have them, and we've got to figure out when and how much.
That's similar to what like Sarah's saying, but also from the standpoint that early studies for in vitro maturation just used follicular. Remember, right? Now we've got all these other ingredients get added to commercial meat. Wondered if there was an opportunity to get follicular fluid commercial media with different ingredients, and even use kind of like the subtractive metabolomics of what's left after those sites do it.
Yes. Ask Hannah in a year. Maybe not at the level you're talking about, but also really interested in seeing what do they use, how much are they putting out throughout with the commercial media versus with follicular fluid supplementation. Maybe add, like you had said, follicular fluid only. I think that's where we've got to go, because we've got to see what they're putting out as well as what they're taking in. And hopefully we'll get some of that out of Hannah's master's and then can continue. And I say Hannah, Tom knows Hannah. Hannah did an internship with Tom before she came to me.
Talked about the metabolites. See the essential as well.
Right.
You're starting to, but you've seen differences of it.
Right. So the only RNA-seq data that I currently have studied or analyzed would be what we did together.
Yeah.
But I do have RNA-seq data from those 3 time periods that we can look at. And I agree with you, that's a longer abundance within the oocyte, because we're not going to have those, those RNAs are going to be throughout maturation, fertilization, and they'll stick around until they're degraded when we have embryonic genome activation. So they're there for a while. It almost makes me wonder too, does this, we think about the oocyte as transcriptionally pretty quiescent by this time, but I think we also know that it's not fully. There's still little bits going on, and then what the cumulus is sending. It just makes me wonder if this environment is inducing some genes that really help later on as well. Thank you.
Dr. Moorey again.
Thank you all.
And just a reminder, grad students' lunch will be down the hall.
It's a rascal.
Just do it. Because I've had it for like